Okay. All right. Well, hello. Welcome everybody to the PhD defense of Ted Bambakidis. We have a lot of people on Zoom and many people here in the room. So the order of things is that we're going to have Ted's seminar and then questions from the public. That's you. I'll be monitoring the Zoom. And then we'll have a closed door meeting with Ted's committee in the front row here. And then at 5 o'clock this afternoon, we'll be gathering at the Downward Dog, where I'm buying drinks. Okay, so let me tell you a little bit about Ted. This is a standard tradition of, so you just have to put up with it. Ted got his bachelor's at the Ohio State University, where he wrote an honors thesis on zebra mussels in Lake Michigan. He worked as a technician for Bruce Peterson at the Marine Biological Labs in Woods Hole. Some of the people online know Bruce Peterson. Doing work at Tulip Field Station in Alaska. Then he earned a and then he was also a research fellow at the Great Lakes Environmental Research Laboratory. Then he earned a master's in stream ecology and benthiology at Bowling Green State University. And then he decided to broaden his expertise by working in the biomedical field for 6 years at the University of Michigan, where he gained a lot of experience in DNA sequencing and omics analyses, and was very productive there, authored or co-authored 18 publications and a book chapter, including one paper titled Valproic Acid Modulates Platelet and Coagulation Function Ex Vivo. Great time. In Michigan, he got married, I think it was in Michigan, and at about the same time in 2019, Simone was accepted to a medical residency or residency at Oregon Health and Sciences University, and Ted was accepted here to work with me. I can't tell you what a pleasure it's been to work with Ted. We did more than a year of intensive field work collecting samples across the whole country for the NSF-funded project we call the Watershed Rules of Life, which is something he's going to be talking about. But there are a lot of things we did that he's not going to be talking about. We wrote several proposals to NSF and the Department of Energy. We co-authored a paper on the role of microbes in stream restoration projects. We analyzed data from an ongoing Big Rivers project that I'm doing with the U.S. Geological Survey that Dylan's involved in too. And we participated in this big DNA sequencing project with Kelly Wrighton and Caleb Borton and some others, PNNL people, Joint Genome Institute people, to build a global database of river metagenome assembled genomes, which is, I think, available now. And along the way, we built an amazing network of colleagues and collaborators all around the world interested in river microbiome science. So working with Ted's greatly expanded the scope of my research and my science, and has taken me in directions I did not anticipate. I feel very fortunate to be working with. So with that, I will hand the microphone to Ted, and he will present his dissertation, the fundamental physical controls on river bacterioplankton.
Thanks for those kind words, Byron. I appreciate it. Some half-truths there and some embellishments. I appreciate it nonetheless. Thanks everyone for coming this morning, live in the room and on Zoom with us. Yeah, let's get started. So here's an outline of what I'll cover this morning. First, we're going to do a pretty brief primer on river ecosystems, just as some background. Then we'll jump into the first project, which looked at controls on river bacterioplankton communities in a large temperate watershed. Then we're going to go to the tropics to ask some of the same questions down there to see how tropical rivers are different or the same as temperate rivers. And then we're going to actually look at bacterioplankton function or omics, and also introduce a new conceptual framework that we developed in our lab. And then I'll wrap up with some conclusions. So over the past decade or so, we've learned that rivers play really an outsized role in global biogeochemical cycles. So our colleagues Xiao Liu and Pete Raymond showed that rivers are extremely heterotrophic systems, and they have CO2 concentrations that are often an order of magnitude higher than atmospheric CO2 levels. And if we look at efflux levels as well, CO2, rivers tend to off-gas massive amounts of CO2, all river systems in every single biome on Earth. They're extremely heterotrophic in terms of net ecosystem production. And if we look at an annual basis, the amount of off-gassing from rivers globally, it's about 2.3 petagrams every year. And put that in perspective, that's on the same order of magnitude as our sinks for carbon in the oceans and in the terrestrial biosphere. So just a really, really massive amount of carbon dioxide and methane being released into the atmosphere from rivers. So let's look at a watershed and try to understand where the carbon comes from and what happens to it in river systems. So river networks connect our distant continental interiors to the ocean, and they have this dendritic structure. So that means that up in the headwaters, there's a lot of terrestrial connectivity between rivers and land. And so that, therefore, we call these allochthonous systems because a lot of the energy and materials are predominantly coming in from the landscape. So things like snowmelt, precipitation, it's kind of flushing in, so to speak, carbon and organisms from the terrestrial landscape into the rivers. And then as rivers combine and flow downstream and become larger, that terrestrial connectivity, it dissipates, it becomes lost. There's just less surface area connected to the terrestrial environment. And it becomes more of an autochthonous system fueled by dissolved organic matter, DOM, from higher up in the watershed. And you also have primary productivity occurring as well in these large river systems. And along that flow path, the dissolved organic matter gets broken down into smaller and smaller substituents by sunlight and by microbes. And we end up having quite a bit of, as I mentioned, CO2 off-gassing, as well as some carbon exported into the ocean. And so biogeochemists tend to call rivers as bioreactors because they're taking this carbon from the landscaping, transforming it, transferring it, then releasing it to the atmosphere or to the ocean. So what are the mechanisms of these so-called bioreactors? Well, river bacterioplankton presumably play a large role in this. But how are bacterioplankton communities in rivers structured? That was really a main question of this dissertation. So several studies in the past from Byron's group and others have suggested that headwater streams have unique communities, but those communities change as we go downstream. So here's a fantastic study by Carla Ruiz Gonzalez and Paul Del Giorgio, looking at a watershed in Quebec and different habitats that they sampled, such as soils, soil water, streams, lakes are different colors. Size of the color is our microbial diversity. So you can see there's a pretty clear gradient here in terms of the organisms, how the organisms change in relationship to their habitat, and also how the diversity gets smaller and smaller as we're sampling along this gradient. And in a separate study, Byron and our colleagues Steve Good and Don Uricki over in Biological and Ecological Engineering tried to see what structures large and small stream communities here in Oregon. So they split up their data into large and small rivers. We just lost our screen here for a second. Maybe we'll come back. There we go. Okay. So, and on the y-axis here are the results of a series of Mantel tests, the correlation coefficient. So they found strong correlations between small streams and these landscape variables, these geomorphic climatic land cover variables. But this relationship broke down when we got to large rivers. There's almost essentially no correlation. There's no association here. And so that makes us wonder, what's going on, right? So how come we have this breakdown, and what is structuring these large river communities then, if it's not the landscape? So to look into this, Byron actually led a study on the 6 largest Arctic rivers, these big river systems, and they were sampled over a couple of years and over several seasons. And here's one example of the results from the Yukon River. And there are 3 distinct seasonal communities that are recurring every year. And all the 6 Arctic rivers exhibited the same pattern, this perennial seasonality. So that's suggesting that there's some sort of strong seasonal component that is likely affecting the bacterial communities in large river systems. So let's revisit our schematic, and now we're going to look at it from a microbial perspective. Okay, so we learned that headwaters are seeded from the landscape via mass effects, and we have this highly diverse community of microbes that are entering the system. Downstream, there seems to be some sort of species sorting or environmental filtering that's happening that's changing the composition of our communities. There's less interaction with the terrestrial environment, and in-stream, in-river processes really seem to matter a lot more. So, we're wondering what causes this shift? What's happening here? So other investigators, as I said, we looked at seasonality, some aspect of seasonality, dendritic distance or watershed area or discharge. Maybe it's interactions with DOM, right? That could be something. So here's what we think, and this is what forms the core of my dissertation research. So there's some aspect of hydrology that other studies have looked at, and we actually think that the primary aspect of hydrology that matters most is water travel time. So there's an emerging property of hydrology, we think, and we think that's water travel time. So we think that travel time is a uniquely important feature of rivers, and that there's probably some minimal amount of time for an organism to be in the water column, where there's a process where a dispersed organism can either adapt and survive in the river water column environment, or they can't, and they perish, and they're replaced by those organisms who can. There's a selection process, so to speak. And travel time is dynamic. It's not a fixed physical property of rivers. It changes with drought, it changes with storm events, it changes day to day, really, right? So second, we think that temperature is an emergent property of what we've been calling seasonality. So if we think back to the Arctic river studies, yes, it's driven by seasonality, but temperature is likely underlying a lot of what we're talking about here that can explain a lot of those changes that we see. So we hypothesize that it's these two physical controls, it's temperature and water travel time, that are structuring river bacterioplankton communities. So before we jump into studies, I want to pause and briefly cover what do we mean by water travel time, right? So it's the average amount of time that it takes for water entering the stream channel to reach your downstream point of interest. And so to compute this, I'm going to go over it very briefly. First, we calculated the flow velocity for each reach in the river network. And then we were able to compute the travel time for that reach in the river network. And then we have our downstream point of interest where we collected our water samples from. What we're able to do with that is at a specific sampling location, using equation 3 here, we can compute the cumulative travel time at that specific point of interest in the river on that specific sampling day. So that's what's really unique about this. We can do it for the exact day that we want to collect our microbial samples. And here's an example using real data from the Willamette watershed. Okay, so first right here, this is a headwater stream in the H.J. Andrews Experimental Forest. The travel time of about 1 hour. That empties into the Blue River. This is above the Blue River Dam, the travel time of about 5.5 hours. And then below the reservoir, travel time jumps up. It's about 365 hours or about 15 days. But then the Blue River empties into the Mackenzie and travel time actually decreases, which may seem a little counterintuitive at first, but that's because, again, we're thinking of almost like this is the age of water. So you actually have other streams entering into the Mackenzie that are diluting that older reservoir water. So that average travel time of a particle of water on the Mackenzie is younger than the water coming out of the reservoir of the Blue River. Then finally, the Mackenzie empties into the Willamette, and when we go and sample in downtown Portland, like I am here, travel time increases again to about 270 hours. So as we see here, travel time is dynamic. It depends on where you're sampling, and there is a very significant effect of reservoirs. Okay, so now that we covered travel time, let's jump into some data. Okay, so for the first study, we worked with Jake Hosen, who's now a professor at Purdue University. Jake designed and led this really massive study on the Connecticut River ecosystem, 30 different sampling locations, sampling over 3 years, all sorts of we targeted the whole spectrum of the hydrograph, so to speak. Storm events, snow melt, drought, everything in between. So it's a really amazing data set. So let's look at some data. So here's a co-inertia plot of our bacterioplankton data. I'm going to be using co-inertias a lot throughout the talk. So let me just pause briefly and explain what they are. So it allows us to take our species abundance data set, right, our ASV table, and our environmental data set, and create one model from them. So here we have a representation in 2 dimensions of our beta diversity between samples, and also our environmental variables. And the direction of those variables indicates that they're associated with the samples that they're pointing to. Then the magnitude of the arrow indicates the strength of that association. So here, what I really want to highlight, this is colored by temperature because temperature is really a very, very strong vector along axis. And here's the exact same data, and now I just colored it by travel time. And there's also, likewise, a very strong association here between travel time and our samples. So to better understand the role of temperature and travel time, we ran linear mixed effects models. So we split up our samples by subwatersheds. So the colors on the plot here correspond to our subwatersheds on the map to the right. And we found that the combined temperature and travel time model had the best overall model fit in our linear models. And then we actually found that the slopes between these different subwatersheds didn't differ. So that's suggesting that there's similar relationships between community structure and temperature and travel time in all of these subwatersheds. And that suggests to us that these communities almost exhibit similar trajectories in their community development across the spectrum of temperature and travel time, and also across this range of stream sizes and flow conditions, whether it's headwater streams or large rivers. So going back to the same co-inertias I just showed before, now I just put them side by side so we can just look at them. We noticed something really interesting. So we noticed that community composition at specific sites would shift with temperature and travel time. So I'm going to show 2 examples here. So if we look at deer circled in black, I also circled it on the map here, and then still, we have a lot more sites for still. Still is also circled on the map. So on the right side of the plots, we see that when temperatures are cooler and travel times are shorter, these downstream sites they exhibited communities that were more similar to headwater communities, what we normally find in the headwaters. And so when that happens, we think what's happening is that these downstream sites contain water that is younger, that's higher, that is higher in the watershed, full of more dispersed organisms. It doesn't have more of a mature or typical bacterioplankton community that we would expect during, for example, base flow conditions or warmer conditions, or what we might find in older water. There's just less time for bacterioplankton to develop. So in the Connecticut River, we found strong evidence that temperature and travel time seem to drive bacterioplankton community structure. And we think that the pace and trajectory of bacterioplankton may be controlled by the time available for a community to grow and change, and the temperature regulated growth rate for microbes in the river. So for our next project, we shifted gears and looked at a tropical river system. And so we wondered what structures tropical river bacterioplankton communities. We had the opportunity to get samples from the Congo River watershed. So first, I want to frame this for you and highlight the importance of tropical rivers. We already talked about rivers in general, but it's worth mentioning that it's really tropical rivers that are the biggest emitters of greenhouse gases. They account for 57% of all river CO2 emissions. And to just also give you an idea of this, the Amazon River, a study found that it emits as much carbon from the water as it sequesters in the terrestrial environment. That's amazing for me to think about. I think it's just an amazing piece of data there. So, and in sub-Saharan rivers emit a petagram of carbon every year, just in sub-Saharan rivers. And again, that's on the same scale as our ocean and terrestrial carbon sinks. So the Congo is the second largest watershed on Earth. It's behind the Amazon. Most of the Congo River carbon is off gas. Most of it is exported to the atmosphere as CO2 or CH4. And a much smaller fraction makes it to the ocean. The tropical rivers are poorly studied. There's a much more poorly studied system than our temperate and even Arctic rivers. So again, part of our interest here is just to discover what sort of microbes were down there and what were in these stream communities. So our collaborators, Paul Mann and Max Holmes, partnered with a Congolese scientist, Bambakidis. They collected 21 samples in a synoptic sampling campaign in 2010. And samples were collected from streams flowing through tropical forest, savanna, and also the Cuvette Centrale, which is the largest swamp forest on Earth, actually. And they collected a wide variety of environmental parameters, stream side and lab, also greenhouse gas measurements, which we'll see is very important as well. And we supplemented some of this with some remote sensing data as well. And I include this photograph in part because Paul related to us that the color of some of these rivers was just remarkably dark, brown, and black. And this was reflected in the DOC concentrations, often an order of magnitude or higher than what we normally see in temperate rivers. It's a very carbon-rich system. Okay, so first on the left, we ran a PCOA of weighted unifrac distances. Sites are colored by travel time, and I circled here our three largest rivers in the system, the Congo main stem, the Obangui, and the Sangha. They exhibit pretty similar community compositions, and so we wondered what might be driving that. So we ran a co-inertia. We see travel time, temperature, and dissolved oxygen are most associated with those sites. And that was pretty remarkable to find. It's sort of in line with our findings from the Connecticut on the importance of travel time, temperature. But we wanted to dig a little deeper and figure out what was structuring the smaller streams in the system, the mid and small streams. So we removed those three largest rivers. We generated a new co-inertia model. We saw a separation of sites along axis 1 here with pH, spectral slope ratio of our dissolved organic matter, wetlands, and DOC. And we noticed that these sites over here clustered together, indicating that they had pretty high community composition. And these were also among the most wetland-rich, DOC-rich, most acidic sites in the sampling campaign. We also had a relatively low DOC spectral slope ratio, just indicating that those microbial communities were associated with higher molecular weight, allochthonous DOC molecules that are recently imported from the landscape. On the right, we included a Spearman correlation coefficient just to show that there are significant correlations between high DOC concentration, low pH, our low slope ratio, and our high wetland coverage. So we also ran indicator species analysis, and that showed that the high wetland group was dominated by a member of Burkholderia, a member of the Verrucomicrobia freshwater group, FUKUN18. But interestingly, we also found a species of Novosphygonium. And that genus is known for its ability to metabolize aromatic carbon molecules. So that's suggesting that maybe if we combine our observations from the spectral slope ratio and the presence of Novosphygonium, we actually have an organism that might be able to take advantage of these aromatic carbon molecules. So, carbon. We also found Rosicarius fermentans, and that's a fermentative alpha proteobacteria. And that's been described in the literature as being associated in the methanotrophic consortium. And so its presence might suggest that fermentation may be an important metabolism here as well. And along axis 2 now, we see differences mainly in greenhouse gases, oxygen, and travel time. So these are a lot of mid-order streams down here, the Alima, the Kuyu, the Lobi. So we have higher DO, higher travel time. The indicator species showed that we had members of Rhizobiales, Burkholderia, and the Planktophilia HGCl clade. That's another really well-known freshwater group. So that's indicating to us that travel time still has some importance in these mid and small size streams, and they contain some typical freshwater bacteria. Also on axis 2, methane and CO2 covariate, and I want to point out that both methane and CO2 were super saturated at all of our sites, extremely, extremely high levels. But these two had the highest levels of CO2 and CH4. Here's some data on that. You'll notice also dissolved oxygen. It wasn't anoxic, but it's pretty low. It's pretty low dissolved oxygen. So we ran indicator species. We found our very ubiquitous Percolaria. We also found methanotropes, right? So we found one taxon of Methylococcales and 2 of Methylophalaceae. And Methylophalaceae in particular is known for its ability to use DOM breakdown products like methanol and methylamine as carbon substrates. The literature showed that it's commonly found in temperate and subtropical hypoxic lakes. So that tends to be sort of in line with our dissolved oxygen concentrations as well. Okay, so here is a relative abundance plot, and the first thing I want to point out is we did see our methanotropes pretty much present in almost all of our samples, and that's sort of in line with our very high methane conditions that we have at all of our sites. So to try to make sense of this, we order a relative abundance plot by increasing water travel time. And we see a dominance of Calmondaceae and other Burkholeriales really at most of these sites. You see the 2 shades of blue here. But aside from that, we see a lot of heterogeneity, especially in these smaller stream systems over here. We think that's likely reflective of just the heterogeneity of the landscape in the tropics. These are small streams, they're over a relatively large geographic area. So we just have maybe a very diverse assemblages of organisms entering these different streams. But we did notice a bit of a convergence in community structure as travel time increased. Really around like Lukala Musaka and then these rivers to the right, we start to get more similar relative abundances of actinobacteria, Methylophalaceae, Bacteriodota, or Microbia. Then the 3 largest rivers here as well, pretty similar bacterioplankton community composition. So the last analysis that we did for this paper was we tried to do a comparison between temperate and tropical rivers. So this is a PCOA showing samples from the Congo River. We actually added in some main stem monthly sites that Bienvenue was processing for us. And also the Mekong River that Aaron Coney collected for us in the upper Mekong watershed. And then we have Connecticut River over here. So we can see almost immediately from the PCOA that our tropical rivers are clustering together separately from our temperate rivers. And so that's suggesting that there may be clear differences in bacterioplankton community composition between tropical and temperate river systems. So the key takeaways from the Congo, we think that large and middle-sized rivers are still driven by temperature and travel time in tropical river systems. They might have those same physical controls that we observed in the Connecticut. But certain habitats can certainly have distinct bacterioplankton communities. We saw that these acidic, DOC-rich, wetland-associated streams harbored very distinct communities. Methane, again, it affects the entire basin. Okay, so it really, and that explains our abundance and part of all these methanotropes. So we did look for the presence of methanogens in our taxonomy as well. We know that's an anaerobic process, of course. We didn't see any because all of our sites had enough oxygen in them, we think, to inhibit the presence of methanogens. But we suspect the methanogens are likely in the river sediments, in the hyporheic zone, in the floodplains on the margins that are periodically flooded, and likely in the wetland soils as well. And the methane that they're generating is diffusing into the water column in the river. And then finally, we found that tropical river communities are more similar to each other than they are in temperate river systems. So that suggests there may be some sort of latitudinal component to bacterioplankton communities. as well. So this is a really exciting project. We're very close to submitting it. A lot of just cool science and novelty in it. Really great opportunity that Paul Max and be the new gave to us. Okay, quick sip of water. Okay, and so here is the final chapter. And this is when, sorry, the final chapter, what we're trying to do here is try to synthesize some of the observations that we gained from the first two chapters. And we're also trying to take a look at river microbiome function. So far in the first two projects, we learned that temperature and travel time, they largely shape the development of bacterioplankton communities. So given that importance, we're wondering, is there some way that we could incorporate or integrate temperature and travel time into some sort of conceptual framework that can be used to predict the bacterioplankton community of a given river reach of interest? And our colleagues demonstrated a similar approach for dissolved organic matter. So Shauda and Pete were able to determine whether or not a given river reach contains DOM that is being transformed, undergoing chemical transformations, or it's being exported downstream. And our collaborator, Kevin Ryan, was able to use that DOM framework to actually predict DOM composition. And so our motivation here is that our first objective was to develop this novel conceptual framework for river bacterioplankton that could predict what was happening to the community composition of a given reach. And second, I've hit on it a couple of times, the importance of rivers in biogeochemical cycles, especially carbon. But remarkably, there's not a very big literature on function. We just don't really understand on a metabolic level what the microbes of these rivers are doing. So using metatranscriptomics and through the lens of this new conceptual framework that we developed, that was our second objective for this study. So to do this, we intensively sampled 4 large watersheds in the United States, the Willamette and Deschutes here, the Gunnison River in Colorado, and a new sampling campaign on the Connecticut River. We had about 11 sampling stations in each watershed. We sampled once a season for 1 year. And we sampled river mouths every month to get some high resolution data on our most downstream site. We collected water samples for stickiness, metagenomics, metatranscriptomics. We modeled travel time. We high resolution dissolved organic matter, nutrient chemistry, and differential gene expression as well. Just a few photos from the field. Here's Pete servicing the dissolved oxygen sensor on our winter sampling in the Moose River. This was like 2 days before lockdown and the pandemic. Here's Byron filtering water off the back of the snow machine. We just had a sample in February up in the headwaters of the Deschutes. We realized pretty quickly the only way to get up there was up there, so a little bit of adventure science. Then barium sampling in the headwaters of the Willamette River. So we call our new conceptual framework the microbial dam cooler. And the dam cooler number concept is borrowed from chemical engineering. And that indicates whether a system is reaction dominant or advection dominant. And so to develop the microbial dam cooler, first we estimated the growth rate for river microbial communities. So we know that biological growth depends in part on temperature. And so we assumed that temperature would affect microbial growth rate according to the Arrhenius law. So our assumption was that for every 10 degrees increase in temperature, we would have a doubling of microbial growth rate. And then the doubling time of the community was estimated using a natural log and then our microbial growth rate. And finally, our dam cooler ratio, it's unitless, it's just a ratio. That was our travel time over our doubling time. And our interpretation was this: when dam cooler is less than 1, it's an export dominant system. And So a way to think about that for microbes, that would suggest to us that it contains organisms dispersed from land. And then when dam color is greater than 1, it's a reaction dominant system. We would consider that to be a more established planktonic community that's adapted to the water column environment of rivers. So let's look at some results. So first on the left, here's a PCOA of 16S data to start out with. And we see that dam color is strongly associated with our samples. And again, these are samples from 4 watersheds, headwaters to mouth. There's not a lot of, they're not really clustering by watershed, but we do see this very strong association with dam color. We ran a quick co-inertia model, and dam color was the strongest environmental factor among all these samples. So from there, we looked at metagenomes and metatranscriptomes. And this is a PCOA of metagenomes and metatranscriptomes to the left and right. Here, it's by watershed. I know that might be hard to see, but the take home here is this. There is not a clear trend here based on color, based on watershed. And we looked by season. Same thing. There's not a really strong clustering or association between season and our metagenomes or metatranscriptomes. But we do see a stronger association with dam color. So we see that the relationship between our metagenomes or metatranscriptomes seems to be much closely related to our dam color ratio than it is with season or geographic location, which we were very pleasantly surprised to see, but really almost shocked by those findings. And again, I want to stress the heterogeneity in the system. So this association held up whether it was headwaters of the Gunnison River or the Gunnison River Terminal Station, all of our sites in between, the Chutes headwaters, End Member Station, the urbanized site in Portland. So this is really a remarkable initial finding. So next we ran differential gene abundance and gene expression analysis in EdgeR. We kept the analysis fairly simple. It's a single factor design. We just separated our samples based on the dam color number, if it was above 1 or below 1. And here we have some numbers here on our differential analyses and a few metrics that we looked at in addition to the classic log2 fold change. We looked at transcripts per million, that's our relative abundance measure. And then we actually looked at transcripts per liter of river water. So we actually used RNA spike and controls that then on the back end after sequencing, we were able to compute on a per liter basis transcripts that we actually found in the river. And so here's a big overview of our metatranscriptomics data. The only data here I'm going to be showing are more significantly differentially expressed. And on the y-axis here, we use KEG for our annotation. On the y-axis, we have our KEG pathways. And I also group them into some just larger categories for readability. And we have box plots showing the difference in log fold change between the two groups. And this convention is going to hold throughout. So when a positive log fold change over here on the left indicates a low dam color number. We labeled it up here too. So again, this is when these are, we think, dispersed organisms recently dispersed from land. A negative log2 fold change corresponds to a dam color greater than 1. So those transcripts were upregulated at dam color greater than 1 sites. So upregulated, low dam 1, upregulated, high dam color. That's the way that we thought about it. So let's start with carbon fixation. So we can see here at a high overview is that photosynthesis, Calvin cycle, and carotenoid biosynthesis pathways were upregulated at our dam color greater than 1 sites, or high dam color sites. And I'm going to be using the same type of figure a few times, so let me walk you through it. So here's our list of genes for photosystems, Calvin cycle, and carotenoid biosynthesis. Left of that now we have dot plots. And the color of the dot plot indicates the change in relative abundance, with more yellow indicating higher relative abundance. And the size of the dot plot is our absolute abundance. So the bigger this is the top line, the greater absolute abundance. But these are means over all these samples in the low dam polar and the high dam polar groups. So we see right away from the dot plot very clear differences in relative abundance and in many cases absolute abundance in our photosystems, Calvin cycle, and carotenoid biosynthesis, the dam polar greater than 1. And here's also a heat map, and I brought that up as well to just show the variability across samples. We have 101 metatranscriptomics samples. Here are dam polar values up here. We also have just some clean cutoffs down here for readability. The take-home message from the heat map is that there's not a ton of variability. We see a pretty consistent increase in gene expression with increasing dam polar in all of these pathways for carbon fixation. Okay, so next we looked at the breakdown of lignin phenol aromatic compounds. Here are the data. And on the heat map, we found a relatively consistent pattern of transcription of enzymes involved in the catechol and protocatechuate central pathways of lignin phenol breakdown. So increasing dam polar, decreased expression. Another way to think about that is that when dam polar was high, these were overexpressed, right? We see that in the dot plots as well. Small dam polar, very high expression of these lignin phenol breakdown enzymes. And so what are catechol and protocatechuate? Well, bear with me. This is a very detailed diagram, but I'm gonna go over it relatively quickly. So lignin is composed. So lignin is it comes from a terrestrial organic matter, right? It's an indicator of terrestrial organic matter. It's typically broken down into several different substituents of aromatic rings, low molecular weight aromatic rings that enter into the stream. Here's an example of 4-hydroxybenzoate. It gets converted to protocatechuate. And for the purposes of this diagram, a red enzyme is significantly upregulated at dam polar less than 1. Here's our typical catechol substituted derivatives as well. So a variety of these small organic molecules. What we saw was an upregulation of these enzymes that can break open the aromatic rings. So up here we have lig A, lig B, lignin, right? Breaking open the aromatic rings. And for catechol down here, cat E is kind of the main workhorse here. It can take a variety of different substituted catechols and break open the ring. It was upregulated, as were these other enzymes as well. And if we carry this through, we see that once the rings are broken up, they get broken down into smaller and smaller and smaller organic molecules, and ultimately into things like acetyl-CoA, pyruvate, which we know are really important organic molecules to get fed into the citric acid cycle and make our intermediates that we need to generate ATP. So the take home here is this: not all enzymes were upregulated. Some of them were even downregulated at dam polar less than 1, but there's a lot of red. So we saw a pretty consistent pattern of upregulation of lignin phenol enzymes when dam polar was less than 1. And so that made us wonder what other carbon chemistry might be happening at dam polar less than 1. So we looked at methanogenesis. We saw significant upregulation of transcripts involved in the subunits coenzyme M and B, which we know are involved in the final stage of methane synthesis. And to dig deeper into this, we looked at the taxonomy, and we found that this was exclusively being expressed by archaea. And so trying to piece this together, we know that this is an anaerobic process. So we suspect that the gene expression that we're seeing from these water samples is remnant expression from these organisms that were expressing it and were originally in river sediments. They were in groundwater. They were in the hyperrheic zone. And that sort of makes sense because a lot of our sites, especially out here, a lot of streams in the Cascades are groundwater fed. So that's what we think we were able to detect here. And this sort of, so between our lignin phenol breakdown data and methanogenesis, it kind of reinforces the idea that at dam polar less than 1, it's an allochthonous system composed of organisms that were recently dispersed into the stream, and they're poorly adapted to the water column environment. And finally, let's look at nitrogen. So first, we found upregulation of genes involved in dissimilatory nitrate reduction to ammonia. Those are genes such as NAT-A, NAT-B, and NIR-B. We saw upregulation of nitrogen fixation genes when dam polar was greater than 1, such as NIF-K and NIF-D. So that would suggest we've got a lot of cyanobacterial activity at dam polar greater than 1 sites. So now let's talk about nitrification. So we found upregulation of several genes involved in a putative nitrification pathway when dam polar is greater than 1. So the first step in nitrification, where we have the conversion of ammonia to hydroxylamine with the AMO-CAB enzyme complex. It wasn't significantly different between groups, but it was present across our samples. So I included that up here. So then the second step, we would have hydroxylamine being converted to nitrite, and that's with the HAO enzyme here. And then we have NARG and NARH finishing up by converting our nitrite to ammonia. So those are all upregulated in our dam polar greater than 1 site. But somewhat confusingly, we also saw NIR-K upregulated at our dam polar greater than 1 site. And we know that NIR-K is involved in the second step of denitrification, the canonical denitrification pathway, where it's converting nitrite to nitric oxide. And so we're sort of scratching our heads and really wondering what's going on. Are we seeing denitrification? Are we seeing nitrification? What might be happening? So we looked at the taxonomy. We saw that NIR-K was being expressed by nitrospiraceae, by our true nitrifiers, right? So that sort of ruled out denitrification. So it's being expressed aerobically. These are all oxygenated samples. And so we looked in the literature, and here's what we think is happening. So first, we have ammonia being converted to hydroxylamine by our AMO-CAB. In the canonical pathway, of course, we have HAO converting it to nitrite and then nitrate. But there's also a putative pathway in the literature where HAO can also convert hydroxylamine to nitric oxide. And then there's evidence that NIR-K can actually work in reverse. So it can actually convert nitric oxide to nitrite. Then we have NARG and NARH finishing off the process and generating our nitrate. So this is a putative pathway. There's some evidence in the literature for it, but our findings suggest that this might be happening at our dam polar greater than 1 samples. Okay, so takeaways from this study. First, we introduced a new conceptual framework for rivers, microbial dam polar, that can predict the status of river bacterioplankton communities, whether it's dominated by organisms from land or truly adapted planktonic community. Found that dam polar is a useful framework for examining changes in gene expression across watersheds and seasons. And this was the first study to look at river bacterioplankton function at this scale. Up until now, haven't been comprehensive studies like this looking at several watersheds, headwaters. Okay, so to wrap up my talk. So this research involves 6 different watersheds on 3 different continents. And it was really fundamentally motivated by recognizing that rivers have a big role in the carbon cycle. We don't know a lot about their structure and their function. So we learned that temperature and water travel time are fundamental physical controls on river bacterioplankton communities, with temperature regulating the growth rate of the microbes and travel time setting the time scale and pace of the development. And we also extended that and developed a new framework, microbial dam polar, which incorporates temperature and travel time to predict our communities. And we also looked at river microbiome function through the lens of dam polar. And our next big step is to corroborate our omics findings with environmental data, with our nutrient data, to actually pair together and build up these networks. So we have our enzymatic data and also our nutrient chemistry data, things like ammonia, nitrate, etc. And thank you for your attention. Thank you to our funders, especially NSF, USGS, very generous support from OSU Microbiology and CEOs. and happy to answer any questions.
Have some questions from.
Great talk, yeah, that was super different. I have a bit of a different question. So I know how challenging river sampling is and how many issues you run into during. And I was just wondering what some of your major challenges were in, say, overall sampling rivers and what main advice you give to future river microbiologists.
So the question for folks on Zoom, the question was the challenges of sampling rivers and future advice for river microbiology sampling. I would say a lot of the challenges are actually sensors. I think that actually in many ways collecting water is the easy part, filtering and dealing with all that stuff, but really all the rivers and even the rivers out here, which are pretty well regulated by Bureau of Reclamation and things like that. When we think about reservoirs, we lost probably $2,000 or $3,000 solid oxygen sensors, and we were battling the whole time about how to anchor them, make them retrievable, things like that. We try to collect light sensors. We had to do light sensors too. That was also challenging. So I think that in many ways, even doing it pretty, elliptically, connecting our and filling up our Starbuckses in the field were pretty good. I mean, Mark's got a very good technique for that he taught me. And I think sensors were the big challenge. That's it. Yeah.
Thanks, that was really fun to hear about. Been curious what you've been doing all this time. So your approach to getting to a generation time was really interesting and cool. And I guess I'm considering the big impacts on CO2 emissions. I'm wondering if you can connect or have any thoughts about where most of those emissions are occurring, upstream or downstream. I guess I'm thinking about bacterial growth efficiencies and sort of how that might shift depending on carbon quality, and then you've got growth rate in there. So just sort of, per liter of water is more CO2, or maybe that's the wrong normalization, but where do you think most of the CO2 is coming from across that watershed?
Sure. So I think classically, a lot of the data out there is from big rivers. And it's saying that it's happening downstream, because a lot of the DOM literature is also saying you're getting these really complex, even more recalcitrant molecules in the headwaters. And it just takes time for sunlight, for the photooxidation, and for like the right types of microbes to attack those molecules and actually get that to happen. But certainly there's respiration happening throughout the watershed as well. So to answer that question, what I think that we want to do, and we'll probably end up doing before we submit this last paper, we do have greenhouse gas measurements for all of our samples, and actually N2O and methane. And so I think that's a really great question. I think hopefully to get to that question and kind of, have it to pair it with our microbial data is to actually see what those measurements are like up in the headwaters, mid levels, downstream. And to your question about what sort of normalization, yeah, that's a really great question. I mean, I think surface area, river surface area has been one in the literature. But yeah, per volume, I hadn't thought about that. On per volume basis might also be really useful as well, almost like a water throughput sort of method to think about CO2 evasion.
Yeah, I mean, I guess especially thinking about that latter part about higher gene expression of your catechol users or whatever, right? I mean, they seem to be in the sort of upper reaches, whatever you call them.
Yeah.
Yeah, I mean, I think about those as being maybe less efficient. So I would have thought, you know, because they need a lot of energy to run those, so I almost would have thought the efficiency would have been lower upstream, but it's an interesting, little bit of an interesting conundrum. So anyway, I'm curious about that.
Yeah, no,
Anyway.
that's a really good question. Yeah, I was a little surprised to see this happening so high in the watershed as well, in some ways, yeah, but a lot of this again might be happening in soils as well.
Yeah,
right?
yeah, they're just
So,
coming out.
Yeah.
I have one other, but I don't want to be a hog.
We have one online too, so. go ahead.
Okay, so my other question was about, so I think the travel time idea was really cool. How would stratification in rivers impact travel time and temperature effects? So, yeah, so I didn't know whether there was any examples of particularly stratified rivers and whether that matters.
I didn't remember to repeat the question. So the question to from Dr. Halsey is about stratification of rivers and how that might affect temperature and travel time. That's a really great question, and that's something that we didn't factor in here. But I do know from the literature that stratification can happen in big rivers, and one that's jumping out to me right now is the Congo actually has endemic fish in like the hypothermion, so to speak, of the river because the main stem is so deep. To my knowledge, that we didn't factor that into where we did this, but you're right, because certainly, especially in very big free-flowing rivers, parts of it aren't just, it's not just a box of water moving downstream, right? There are pockets where you could have much higher residence time, much deeper, and certainly where the temperature would be much, much cooler, even in the tropics, presumably, than further up. So the short answer is that we didn't factor that in, but that's a really great idea, and that's something that maybe we should, as we hopefully keep revising and improving the sort of modeling that we can try to build into it.
All right, I have a question from Julie Alexander online. Excellent job, congrats. I wondered if you would comment on the potential implications of the clustering of sample points in your third chapter for a couple of the river systems. Clustering of sample points for a couple of your river systems. It might have been those ones that are off. I think it's the Olmix ones.
Up here, the Olmix ones.
Yeah. Noticed on the map. Okay, so effects on your models, autocorrelation. Just curious if you had thoughts on whether stratifying sampling would improve or not affect the conclusions. Noticed on the map. Oh, maybe it was clustering on the map. Like there were groups in the Connecticut up high and Connecticut down low in the watershed.
I see.
Yeah, so like
Here.
three things.
Yeah. Excellent. Yeah, that's a good point. Is that there is a pretty significant geographic distance between our headwater Connecticut sites and some of our end member sites here, although we do have a smaller subwatershed over here. So the question is, sorry, can you repeat it one more time?
Potential implications of the clustering of sample points for a couple of the river systems, does that affect your models? Is there autocorrelation? Sure.
I think that the way that we developed the sampling design was that even sites that are clustering together on the map here have pretty different hydrology. So, for example, here in the Deschutes, we have a lot of clustered sites in the headwaters. Some of these are free flowing, some of them are severely reservoir impacted. And part of the study design for this project specifically targeted reservoirs to almost have this comparison in the Deschutes and even the Willamette, for example, where we have free flowing rivers and reservoirs in the same geographic sort of areas high in the watersheds. We haven't done a deep dive into this in terms of comparisons beyond what I showed today. But there is a possibility for us to certainly dig deeper in there and see the impact of reservoirs on these otherwise very similar landscape driven headwater streams. I'm not sure if that answers the question.
Great answer, thank you.
Hi, thank you so much for your presentation. There is a part in the first section I was a little confused by. You brought up the Shannon index, and from what I think I understand, you're saying that like upstream there's lots of diversity, like the number of species, and goes downstream there's like less species. And then you brought up relationship between like travel time and temperature. So did your study indicate that those two factors impact species diversity or species composition? Like,
So
were you looking at like, sorry, the total number of species, or like the difference between species groups? Like, I guess that's.
sure. So I think for this study, for the Del Giorgio lab, I think what they're trying to show here is that it's both on this plot. That's my interpretation of this plot, is that the composition is shifting. Right? We're seeing a separation of samples based on habitat, and also the diversity of those samples is decreasing. And that's sort of what we found in our lab, kind of separately, but also on large scale rivers and small streams as well, is that we're seeing just such a diversity of organisms coming in from the landscape that you kind of have this almost streamlining by the time you get downstream, and you have what, and I don't want to call it in an ecological sense like true plankton, but we have more what I would call a smaller group of organisms that are classic freshwater river planktonic organisms, and also organisms that we see in lakes sometimes as well. So there's a decrease in diversity and a change in community composition.
Okay, so you're looking at both.
For our study, yeah, I didn't provide diversity numbers here, but
Okay.
yeah.
Okay, thank you.
That's the last question online. Any final questions? All right, let's thank Ted. Okay, so we have Pete online and our committee up front. Let's take a few minute break. Okay. You can jump in for the rest of us. Thank you, everybody online.
Really, and then.
Yeah.
Okay.